Hello, welcome to the last video on the topic of molecular biology, which will be photosynthesis, a very important process which is mentioned in almost every test ever in biology. So yeah, first of all, I'll be giving a definition of photosynthesis and the photosynthetic reaction, as well as the spectra involved in it. Then I'll be talking about the Engelsmann experiment, the light independent and dependent reactions, as well as its limits and influence on of photosynthesis on Earth, the atmosphere, oceans, and rocks. Hope you enjoy. So first of all, photosynthesis is the process of converting sunlight, carbon dioxide, and water into glucose and oxygen, and it's basically the light energy captured by photosynthetic organisms that have carbon compounds within cells. Light is also composed of a range of wavelengths ranging from 400 to 700 nanometers, and those are also separated by a prism from violet and red. And the object's color is also determined by a wavelength of which is reflected by the light. So as you know, black color absorbs light and white color reflects light. And in the case of chloroplasts, for example, chlorophyll absorbs red and blue and reflects green. So here we have the formula of carbon dioxide and water photosynthesizing to glucose and oxygen, and this is the energy for production of carbohydrates from carbon dioxide fixation, which is something I mentioned in my previous videos if you like to check that out. And the glucose at the end is stored energy for non-photosynthesis, and the oxygen at the end is a form of photolysis, which is the splitting of water molecules. And here we have the absorption spectrum and the photosynthetic action spectrum. You can see these fainter lines in the background. Those are the absorption spectrums of different organisms, mainly here different chloroplasts, carotenoids, with different pigments in relation to the wavelength of light divided by nanometers. And then we also have the photosynthetic action spectrum, which is the rate of photosynthesis in relation to the wavelengths of light per nanometer. And this can be limited by factors of temperature, light, and carbon dioxide concentration, which I'll be talking about soon. Here we have the conclusion of the Engelsmann experiment, and Engelmann was a German botanist living from 1843 to 1909, and here it said that oxygen is evolved during photosynthesis, and different wavelengths of light affect the rate of photosynthesis. So after a period of darkness and oxygen deprivation, motile bacteria, Engelmann used Pseudomonas, they move towards areas with higher concentration of oxygen around the chloroplasts of the algae. So the conclusions were that chloroplasts absorb red, and the photosynthesis and the photolysis leads to oxygen production, and also that bacteria move to chloroplasts because of the high concentration of oxygen. And moreover, it was also said that chloroplasts cannot absorb green, wherein they reflected. There is no high concentration of oxygen there because there is no photosynthesis. So yes, those were the main conclusions of the Engelmann experiment. Now moving on to the reactions, and the first stage of that would be the light-dependent reactions, wherein chlorophyll absorbs light energy, leading to ATP production, and this also leads to photolysis that I mentioned before, where the water is where it splits water molecules into hydrogen and oxygen atoms, and those hydrogen ions and electrons and oxygen, of course, are released. The oxygen is a waste product of photosynthesis and is vital to sustain aerobic organisms due to aerobic respiration, which was the last topic of the video, so you may check that out if you want. And the end of the production is also ATP, water ions, and electrons that are necessary for the light-independent reactions. And those reactions here, which is the second stage of the photosynthesis reactions, where these ATP, water ions, and electrons are taken, and as well as carbon dioxide from the environment, making a range of organic molecules for the plant. And there is also when carbon fixation takes place, so the conversion of inorganic carbon dioxide to organic molecules, plus winning ATP for the process. So we can also measure the rate of photosynthesis. The biomass of the plant increases because glucose is used to produce other plant materials, and aquatic plants release oxygen as they photosynthesize, as most plants, and the volume of bubbles that come out of the plants are measured over time. And aquatic plants remove carbon dioxide from the environment, so the pH value of the water rises, because carbon dioxide dissolving molecules makes a weak acid. And they can monitor the change in pH value over a period of time. Terrestrial plants may do it as well, as they also remove carbon dioxide, but it's difficult to measure as we do not have those water bubbles. And they supply but this is possible by supplying a confined plant with radioactive carbon dioxide, which may then be measured. And you can also determine the biomass at different times with an indirect method. So you have samples that are collected and measured at different times, and the rate of increase is calculated to determine the range of photosynthesis, which is how the measuring of the photosynthesis rate may come to be. We also have three different limitations to the photosynthetic rate, or the photosynthetic action spectrum that I mentioned before, which is that those environmental factors, for example, temperature, light, and carbon dioxide concentration, may make it so that photosynthesis can't increase beyond certain limits, and those can be measured with one variable each. So as you can see, the temperature, because of the energy of molecular collision, increases until denaturation of proteins. The light intensity also reaches a plateau, because at some point, as I already said, they cannot increase beyond certain limits. They're saturated with light. And then also carbon dioxide concentration, which is a vital raw material, but at a high concentration it plateaus, as well as the light intensity, because it's saturated. And next up we have something quite interesting, which is the influence of photosynthesis on Earth, atmosphere, oceans, and rocks. And the first oxygen-producing photosynthesis evolved about 2.4 billion years ago, and this reduced the carbon dioxide content of the atmosphere and also started oxygen. And oxygen is taken up by the Earth's mantle, where oxygen plus red iron makes iron oxide through the oxidation of the iron. And 1 billion years ago the mantle got saturated and with oxygen, and the oxygen remained actually in the atmosphere, and the free oxygen built up as a sufficient support for life, which is then the start of the biological era as we know it today. And consequences of the influence of photosynthesis were that the level of oxygen reached 1% of our current level, and metabolism of eukaryotes started about 2 billion years ago, and there was a greater availability of oxygen that led to many metabolic pathways that were more efficient, and aerobic organisms proliferated in oceans. So carbon dioxide was removed from the atmosphere and went into shells and decomposed sea creatures. This built up marine sediments and became incorporated in the sedimentary of the earth, and later metamorphic rocks. And this may also be seen in the topic of climate change, where the sedimentary is opened or is set free throughout the time, and leading to ocean acidification and further greenhouse gases. Oxygen is also accumulated in the upper atmosphere, so you have sunlight that's acted on and formed ozone. This ozone layer maintained more stable temperature, and the organisms may then colonize the Earth, the land, because of the stable temperature, which was about 400 million years ago. And 550 million years ago, oxygen levels reached today's oxygen levels, and this led to rapid evolution of higher life forms, such as humans, Homo sapiens, and the atmosphere has been maintained by the process of photosynthesis. So it's quite an important biological reaction. And I thank you for listening. These are my sources, and I hope you have a nice day.